Good evening, and I say make I welcome now back to our channel today. Now, this is now the concluding part of the three-part series where we've been on to discuss the bacterial cell and all the structures and functions where they inside different things where they for the bacterial cell. Now, this third part we'll talk about what they call the internal structures of the bacterial cell. We don't discuss the external ones, the middle ones, and the internal structures. Now, if you enter inside the bacterial cell, waiting day for the internal structures of the bacterial cell, the first thing we will talk about now the cytoplasmic membrane. Now, we don't talk about before for the other videos, so make I just talk say this cytoplasmic membrane get wetin they call phospholipid bilayer. Now the structure will be make up this cytoplasmic membrane. Now wetin be this phospholipid bilayer? The phospholipid bilayer contain two major things, and a phosphate head and glycerol tail. So you go see the structure. I go put the structure on top for the screen, make you see am. Say we get wetin they call phosphate head and the glycerol tail. Now the phosphate head dey hydrophilic, meaning say e get affinity for water. Dey attracted to water. But the tails dey hydrophobic, meaning say they no like water and they no wan attach themselves to water. This structure na wetin dey for inside the cytoplasmic membrane, and e dey important to know. Now, apart from this cytoplasmic membrane, e get wetin we dey describe as protoplast. And wetin protoplast be? Protoplast na simply when you look at the combination of cytoplasm and cytoplasmic membrane. Na wetin we dey talk together, gather together, we call am the protoplast. Now, apart from protoplast, the next structure we will discuss na the cytoplasm itself. Some people dey call the cytoplasm the liquid part of the cell. Well, they no dey wrong, but e dey important to note say the cytoplasm get three major structures wey dey together wey we go discuss one by one by one. Now, the first major structure na we will call that area the granular area. And granular area, this na the area wey dey contain many different things wey dey for the cytoplasm. Things like our RNA, our protein, and different maybe lysosomes, ribosomes, all these different things dey for inside the granular area of the cytoplasm. So one dey important. Now, apart from the granular area, we also get the liquid area of the cytoplasm. Cytoplasm get liquid wey dey help dissolve all the substances wey dey for inside the cell. So the liquid area of the cytoplasm dey. Now the third part na the chromatin area. Chromatin area na wetin they call that third part. And wetin that one mean na say that e get a region wey dey for inside the cytoplasm wey dey carry all the genetic material. All the genetic material wey the bacterial cell get, e dey for that third region. And we dey refer to that region as the chromatin region. Now, if we talk finish about the cytoplasm, we'll come discuss the final part for inside the bacterial cell wey be the nucleoid region. Now, we know say bacteria no dey carry nucleus, so e no get membrane around its own nucleus. Wetin e get na just area wey a lot of DNA, a lot of genetic material bin dey that area. And we remember say we don discuss say this bacterial cell get wetin we dey call one single circular chromosome or one single circular giant DNA. So this single circular giant DNA dey coil for that area wey we dey call the nucleoid region, and e dey there, e dey carry all the genetic composition of the cell, all the different genetic information wey the bacterial cell get, dey for inside that nucleoid region. Now, apart from nucleoid region, another important thing to note na say bacteria dey go into wetin we call spore formation. Now, why bacteria go form spores? Bacteria dey form spores if conditions no dey too okay, if the environment dey harsh, maybe water no too dey, the place dey dry up, the bacteria need to protect itself. The bacteria dey go inside wetin we dey call hibernative stage, or we go call am vegetative state. And for this state, wetin go happen na say the bacteria go shut down most of the processes, most of the biochemical processes wey dey occur for inside the bacteria, go shut them down, come create little small cells for inside that bacterial cell. And those small things we dey refer to as endospores. Now, sometimes bacteria dey get wetin we dey call exospores too. And na say if bacteria wan disperse itself, sometimes e go create little spore wey e go push out from the body of the bacteria. That spore go dey outside for the tip of the bacteria, and from there e go find im own way go dey reproduce itself. So e dey important to know say bacteria dey get both endospores and exospores. So make I just do quick recap. All the structures, the internal structures wey we dey talk about wey dey important na the protoplast, the cytoplasmic membrane, the cytoplasm itself wey get cytoplasmic area wey we divide into two. Granular area dey, liquid portion dey, and then chromatin area dey. Then we get nucleoid region of the cell. We don't talk about spores to endospores and exospores. Thank you for listening to this video and have a wonderful day.